mRNA or messenger ribonucleic acid vaccine technology is a phenomenal medical feat that we have recently accomplished and is a crucial player in our battle against the COVID-19 virus that we have been dealing with for over 2 years now. But how does a relatively new COVID-19 mRNA vaccine developed by Pfizer BioNTech and Moderna actually work? Well, that's what you'll be learning in this video in 7 simplified steps. In step 1, the COVID mRNA is injected intramuscularly into the body, usually through the deltoid, or in other words, the shoulder muscle, using a syringe, and this is the only step that can be observed externally. In step 2, we go inside the body where we see the COVID mRNA wrapped in a coating made of lipid or fat called a lipid shell or lipid nanoparticle, enter the bloodstream, also known as systemic circulation. Next, the mRNA is transported by blood to myocytes or muscle cells for the translation of its genetic code into the COVID spike proteins that can be observed on the surface of the actual coronavirus. Step 4 is quite a big one, so I've broken it down into a few substeps so that it can be understood better. First, the lipid nanoparticle attaches to the surface marker of a muscle cell, and this essentially serves as a key to enter the cell, similar to how the coronavirus enters cells in our lungs once we're infected with it. Now that the COVID mRNA is inside the myocyte, it is released by the lipid nanoparticle, which is broken down in the cell, and latches itself onto a ribosome. Now this is where the magic happens. This is where the translation of the COVID mRNA genetic code into the spike proteins of the coronavirus occurs. In this crucial step of the process, the mRNA moves from right to left along the ribosome, and as this occurs, there are these transfer ribonucleic acid or tRNA molecules that transfer amino acids to the mRNA. The anticodons on the tRNA molecules match with codons on the mRNA molecule, creating a sequence of amino acids that develops into a polypeptide or an amino acid chain. Afterward, the polypeptide is folded by proteins in the cell into the functional three-dimensional conformation of a COVID-19 spike protein or antigen, leading us into step 5, where we see the COVID antigens being secreted by the myocyte following protein synthesis. So, now that the COVID antigens have been produced, how does our immune system produce antibodies and develop immunity against the coronavirus? Well, step 1 in this process is antigen binding. So at first, immune cells called B lymphocytes, which are mainly responsible for producing antibodies, do not recognize the COVID antigens as they are entirely novel to them. Therefore, the receptors on the surface of B lymphocytes cannot bind to the antigens. This is where the helper T cells come into play and send chemical signals to the B lymphocytes, which results in a conformational change in their receptors, consequently allowing them to bind properly to the COVID antigens. Following this event, B lymphocytes differentiate into plasma cells that produce anti-COVID antibodies closely modeled after the receptors of the precursor B lymphocyte. Now that the plasma cells have produced antibodies designed specifically to neutralize the coronavirus, if the body is exposed to the virus, the immune system will react immediately by releasing these neutralizing anti-COVID antibodies to protect the body from severe illness and death. Thank you for watching.